Muthu?
Yes, stream start. I said now we can start.
Yes, sir. So, hello. I meant to say the applicants and the researchers and students who are working towards synthetic biology and the people who want to know more about synthetic biology. Here we have Dr. Vijay Singh from Industrial University, and who is going to give a talk to us about the synthetic biology, how it has been present in our country, and how it can be used for different modes of research. So I would like to ask Mr. Muthu Kamaran, so who is going to introduce Dr. Vijay Singh to all the participants here. So I would just ask Muthu now. Please, Muthu, you can start.
Thank you, sir. So every morning has a
Muthu,
new. Yes, sir. Sir, can you hear me?
Yes, Madhu, we can hear you. You can proceed now.
Thank you, sir. So every morning has a new beginning, a new blessing, a new hope. With that note, I am Muthu Kumaran on the behalf of Society of Chemical and Synthetic Biology. Wish you all a cheerful Sunday morning. I deem it a great privilege to welcome you all to the national webinar on engineering of microbial cell factories for production of biomolecules organized by the Society of Chemical and Synthetic Biology. With your all, I would like to recite a शायरी. खुद ही नाप लेते हैं बुलंदी आसमानों की, खुद ही नाप लेते हैं बुलंदी आसमानों की, परिंदों को नहीं तालीम दी जाती उड़ानों की. महकना और महकना तो काम है खुशबू का, महकना और महकना तो काम है खुशबू का, खुशबू नहीं मोहताज होती कदरदानों की. With this shairi, I would, on the behalf of society, I feel delighted to extend a cheerful welcome to our speaker, Dr. Vijay Singh, Society Associate Professor, Dean Research and Innovation, School of Bioscience, School of Science at Industrial University, Gujarat. Prior to this, he was a postdoctoral fellow in the Synthetic Biology Group at the Institute of Systems and Synthetic Biology from School of Energy and Chemical Engineering at the Ulsan National Institute of Science and Technology, South Korea. He was also an assistant professor in the Department of Biotechnology at University University, India, and assistant professor in medical science and biotechnology at the Institute of Advanced Research. He received his PhD in biotechnology at National Bureau of Fish Genetic Resource, Uttar Pradesh Technical University, India. He has designed and characterized a number of synthetic emulators and biological gate gene networks, lycopene pathway, MAGE, CRISPR-Cas9 systems in E. coli. His laboratory focuses on construction of novel biosynthetic pathways for chemicals. Additionally, he is developing an auto-inducible CRISPR-Cas9 tool for targeting genome editing and regulation for programmable removal of bacterial pathogens. He has published in 68 journals, 25 books, 48 book chapters, and 4 patents. He serves as an associate editor and editorial board member and reviewer of a number of peer-reviewed journals. It's all our pleasure to have you with us today, sir. The delay, let's get started. So I request you all to empty your cups and benefit from the session.
Dr. Vijay, you can start, sir.
So, like,
Yes, sir. Yeah.
okay. So, thank you very much, Dr. Karthik and Dr. Vishnu, for giving me the such a wonderful opportunity to present my work here and nice presentation of Muthu. And I'm very delighted and very happy. To share what we are doing and what we did previously. So now I would like to talk on the engineering of the microbial cell factory for the production of the biomolecule because of the petrochemical based it is going to significantly depleting, and we need to find out the alternative or renewable sources to fulfill the current demand through the biological because of the non-degradable nature of the number of the chemical. And now with this microbial cell factory, we can redesign, we can assemble the novel pathways to make this sustainable compound for the nutraceutical, for the pharmacy, for the chemical, for the industry, for the therapeutic. So there are the number of application with this come, and we now have very limited number of the microbial cell factory, and we need more and more cell factory that can produce the chemicals apart from the fast growing demand of the chemical. So we need the land is going to be significantly less because of the construction and other things is coming. And the microbial cell factory, this is the one way, an alternative path where we can make the sustainable production of the chemical uh, to the society. So in this presentation, I will talk on the what are the synthetic biology we did before, and what are the genome editing and the metabolic engineering we are working on the area. So as you know, synthetic biology it's a very new area for India, but it has already 22-year-old. So this 22-year-old, there are the number of the things has been happen in the area of the synthetic biology. So it can be defined as the design and construction of the new biological part, device and the system, or redesign of the existing system for the useful application. And the ultimate goal of the synthetic biology is the create and produce the energy, clean the environment, produce the therapeutics, foods, maintain and enhance the human and animal health. That is the ultimate goal. And as we know, we have the inspiration from the synthetic biology, which is coming basically from the engineering side, and now it is coming into the biology. So this is the basic concept where in engineering we we see there are the number of the part, and that part it can be assembled together to make the device and finally make the system that is running car. In the similar way, the synthetic biology have the small component like the promoter, ribosome binding site, transcription terminators, coding DNA sequences, degradation tag, transcription terminator, non-coding RNA. So these are the part which is not well characterized, not well established, but still we have the limited number of the part. Once this part we can assemble together, make the devices, and we can modify, we can inbuild these devices or install these devices into the microorganism. Then the microorganism have get the ability to utilize the cheaper carbon source, process the information, produce the medicine, chemicals, fuels, or it can sense if there is the some contamination of the air pollutant is there. So this is the way how we are going to move. Still, it's require huge work in this area to make the more standard part like the engineering or computer inside the biology. So this is the important biological part, and this part is vary from the organism to organism. If you talk about the E. coli, a prokaryotic system, we need to have the library of the promoter. It may has the different strength. So if you are making any of the device or system, then we need to have the different kinds of the promoter strength in order to low production of the protein or high production of the medium production. So we definitely need the library of the promoter, we need the library of the transcription factor, we need the library of the ribosome binding site where the gene expression can low or high. It depends upon also the ribosome binding site, and we need the degradation tag, transcription terminator, and the non-coding RNA that is helping for the gene regulation. And we uh, ma- identify this for the bacteria, E. coli, and the cyanobacteria, and it even the bacillus, even the yeast. So number of the standard part is now available. If you want to make any of the device or system, you need to assemble this part together like promoter, ribosome binding site, coding DNA sequences, and the terminator. And then your system will be worked properly, and you will get your protein. So this is the two paper that established the synthetic biology as a formal field, that is the is a synthetic oscillator and the toggle switch. This is the two paper was published in 2000. So it's 22-year has been over, and we make a lot of progress in this direction. Based on that, they designed the synthetic oscillator, which is three gene based oscillator. So there are the three transcription factor that it are like I and the lambda C1, and they have made their own correspondence promoter. So each gene is repressed, and the output it
Dr.
has.
Vijay, sorry for disturbance, sir. Are you changing the slide, sir?
Yeah, I'm changing.
But it's not changing on the this thing, sir.
Really?
Yes, sir.
So
So if you just press in full screen and then you can start. Hello. So just press F5. Then you can put.
See now.
Now it's okay, sir.
Okay. See.
Now it's good, sir. You can proceed.
Okay. So here they, because sometimes the internet bandwidth maybe is too poor, so sometimes it's not coming. So anytime if you see you cannot see changing, then please let me know. So I can stop and again we'll see. So here they made the repressor, that is the checked by the green fluorescent protein, and they find out the cells around 40% was oscillating with the oscillatory period of the 160. And there is the another oscillator made in 2008 by the Jeff Hasty group in the California University's San Diego, and they have identified and made the oscillator that was oscillating with the 58 minutes, and the 99% cell was the oscillating, and that was the one of the best oscillators. So in the right side of the picture you can see the cell is
लोग सर
growing on the
यूट्यूब लिंक
microfluidic
चैट बॉक्स में
chip,
पोस्ट करें सर
and they are oscillating clone, and you will see 99% cell was oscillating. So they
हाँ
made the
ओके
chip, and
पार्टिसिपेंट्स हैं
based
पनीर
on
बना रहे
that they analyze
हैं ओके
and
ओके
they
सर
found out
ओके
that if you put the inducer
ओके
molecule, then oscillatory period can be minimized. So
ओके सर ओके
the best
थैंक यू
oscillator ever exists is the this one. So it has the activator and the repressor gene together. So this is the two gene based oscillator, and the first one is the three gene based oscillator, and this is the one of the good oscillator that was published in the Nature. Based on that work, we decided to make the single gene oscillator, because that time there is no single gene oscillator available in 2009, and we work on the two single gene oscillator, and we constructed the circuit that is going to repress by the black eye theta. So this is the two different transcription factor we choose, and we make the construct like the promoter, black eye, and the YFT we choose, because this is the for the monitoring of the oscillator we need to have. And we successfully constructed the single gene oscillator, fourth single gene oscillator, and we found out our oscillator work properly, and we found 80% cell oscillating with the period of the 180 minutes, and we use the different concentration of the inducer molecule to fine tune the oscillatory period, and we found 90 minutes oscillatory period. So that was one of the good oscillator we designed based on the single gene. Before there are the three gene and the two gene. So this is our first single gene oscillator, and we use the MATLAB to create the heat map, and we identify the oscillation in the different cell was the different. So we saw it's showing in the heat map this cell is oscillating. Based on that, we use and for this characterization we use the microfluidics chip, where we design the microfluidics chips. It has the four different core, one for the media, one for the cell, and the two we use for the waste. Because when the cell is growing on the chip, it is going to be on the waste, otherwise it's made the mismatch. So we design a chip, and we couple this tube with the tubing, and we measure under the time lapse microscopy, and we found out that our oscillator was perfectly working in the microfluidic chip. Based on the single gene oscillator, we couple two single gene oscillator together, and we identify that our oscillator was properly working and independently on the cell, and we use again the microfluidics platform to long term measurement of the single cell, and we know the cell was independently working, both the oscillator was independent, and it can be fine tuned, it can be switched off if we use the one inducer molecule. So this is the oscillation work we did, and as we know, the small non-coding RNA is very important component into the genome that is going to regulate gene expression. So the small non-coding RNA not only playing the positive role for the gene expression, it has also playing the negative roles. Means it can replace the gene function. So as we know that
Dr. Vijay,
yes.
sorry for disturbing again. So again, the slide is not moving. So please, can you put the screen into full screen? Press F5.
Is this okay?
Uh, I think you have pressed, but it's not opening now. I think some technical problem is happening.
Again, you cannot see.
No, sir.
Wow.
I think we are on coupled oscillator screen. Now it's detection of small non-coding RNA. Slide number 11.
Yeah, I can see.
So just put it into full screen, sir.
Can you see? No.
No, it's not. It's still like that only, sir.
Wow.
I think some technical fault. If you just you can roll up again regularly, sir, that will
Can
work.
I put stop sharing, or again I can share?
Yes, yes, sir, you can share again, sir.
Is it visible
Sir, it's visible, but it is not in full screen.
now?
No, sir, it's still like that only. Just try to move the screen as soon as like that, sir. Then it will be okay.
Can you see this one? Okay. Or
Now it's okay, sir, but it is not full screen.
yeah, if I put full screen, then again it's not moving, I think.
Yes, sir. So you can
Wait,
just continue, sir. Just
can you
change the
see?
slider regularly, sir.
Can you see now?
Yes, sir, I can see.
Full screen?
No, not in full screen, sir.
Wow. So something is happened. So can I proceed with this
Yeah,
small?
yeah, please, sir, please.
Okay.
Just change the slide regularly, sir. That's all.
Okay. So the small non-coding RNA is playing very important role in the gene regulation. As we know, the single once the mRNA is formed, it is going to sometimes cis repress, means the ribosome binding site is not available because of the complementary reason. In that time, the small RNA, which is 24 single base long, and that is going to bind with the upstream region of the ribosome binding site on that mRNA, and it can pull out, and it makes available the ribosome binding site. and it then once the ribosome binding site available, it can bind with the ribosome and it can translate the protein. So that is the positive regulation of the non-coding RNA. And the second one is the negative regulation. So the small RNA, this is the small RNA like 24 25 bases long, and it can bind on the uh, mRNA, and it can repress the gene function. So this is the one way. And the second mechanism of the negative regulation, it can bind with the mRNA and it can degrade it with the ribozyme activity. So this is the two mechanism as we now know which is present in the natural form. So in 2004, the group of the Jim Collin in Boston University, they made this basic concept of the non-coding RNA and make the synthetic riboregulator. So the small RNA, it can play the role of the regulation. So that is they put the name of the riboregulator. So it was the first time it came into the existence, the riboregulator term. So as we know, the prokaryotic gene expression, it has promoter, ribosome binding site, and the GFP. Once the transcription happen, it can make the RNA, and this RNA have the ribosome binding site as well as the AUG as a start codon and the full GFP, and it can transcribe and make the protein. This is the prokaryotic system. So they made the riboregulator, artificial riboregulator, based on the promoter, cis repress region, ribosome binding site, and the GFP. Once the transcription happen, then it can make the mRNA with the cis repress region, ribosome binding site, and AUG as a start codon. Once this mRNA is going to form the secondary structure, there is no any translation happen, so there is no protein production. And they have designed the trans-activated RNA that can bind with the cis repress region, and then they allow the ribosome is free and it start the protein expression. So this is the new mechanism it has developed based on the existing nature of the non-coding RNA in the system. So based on that work, we designed the AND gate function. So where we use the two different small RNA that express under the control of the PL-LAC promoter and PL-TETH promoter, that is regulated by the TETH-R and the LAC-I transcription factor. And the inducer of that LAC-I is the IPTG, and the TETH-R inducer is the anhydrous tetracycline ATC. And we use also the constitutive expression of the superfolder GFP molecule, and we use the untranslated region where it make the secondary structure. So once the both small RNA is function, it's active and express, then only that time it can make open the untranslated region of the GFP and it's give the expression. So in the graph during the characterization, once the IPTG and ATC is not available, then the expression you cannot see a lot. So once the so it's like 00 is the 0, and 10 is the 0, 01 is 0, and 11 means IPTG and ATC both are available, then you will see the output. So this is the truth table, the all the logic gate follow, and based on that we made the logic and logic gate in the equalize system, and we chose this is the faster system because it doesn't require the protein. So if there is no protein, then the faster dynamic can get is and less metabolic burden, and it's very tight control system, and it gives the new function of the different way of the gene regulation. And we choose the bacteriophage because as we know we are facing a lot of challenges of the multi-drug resistance bacteria, and some of the bacteria it show the resistance of the fourth generation of the antibiotic. So it's very difficult to treat that type of the bacteria. So we started work on the T7 bacteriophage, that is normally bind with the E. coli and they can deliver the genetic material inside the cell, and it can assemble together after the transcription and translation process and make the new phases, and it can lyse the cell. So we chose the T7 because it's the well characterized and the genetic tool is available, and it the multiplication of this T7 is 17 minutes, so it's quite fast growing phase. So here we engineer the T7 and we deleted the GP5, which is the DNA polymerase gene present in the T7 bacteriophage, and we deleted from there, and this GP5 gene we clone under the control of T7 promoter and transform into the E. coli. So once the GP5 is not available, then the T7 bacteriophage cannot replicate. So here we have the movie actually. The if we put the full screen, then only can be visible. So the E. coli without the GP5. So once we infect phase to the E. coli. So here it cannot attack and not lyse, but the E. coli with the GP5 genes, because of the it support the T7 to grow there, and then it can lyse the E. coli. So here we show that how the T7 bacteriophage kill the E. coli bacteria. So based on that, any of the pathogenic organism, any of the pathogenic bacteria can be used as a technology for the as alternative technology for the antibiotics for treating that type of the infectious disease. So this is the regular work we did in our laboratory, and now we move to the CRISPR-based genome engineering, where, as we know, the CRISPR is the adaptive system where the bacteriophage is attack on the bacteria, and the DNA, especially the spacer region, it is going to integrate into the cluster of the CRISPR array, and it's make the repeat spacer region. Once they are going to transcribe, and it can produce the pre-CRISPR RNA that is going to process and make the mature CRISPR RNA, that is called the adaptation. Once the spacer is inserted into the genome, and after the processing, the biogenesis and processing, it can make the mature CRISPR RNA that can interfere the incoming viruses, and it can degrade. So once the system is available, the next infection with the same bacteriophage is not possible because it can easily kill them. So that is called the adaptive immunity of the system, and now the CRISPR is extensively studied, and still it has the two classes, one and the class two. So class one is quite complex class where it's require number of the protein to make the interference with the system. But the class 2, it has only the single protein that is called the Cas9, Cas12 and the Cas13. So this protein is a multi-domain protein, but it's a very simple mechanism is developed, because if you use the class 1 any type 1, 3 or the 4, so it's required number of the protein for the genome editing. So here the Jennifer Doudna group, they make this Cas9 based genome editing. It has the single protein based function, and it's required definitely the small guide RNA to make its function. So what are the mechanism of the CRISPR, how it can work? So in order to make any CRISPR system, you need the expression of the Cas9 protein and the small guide RNA with the handles that where the Cas9 can bind, and you need the PAM sequences, which is the small region, which is small nucleotide, and the guide RNA can target where you want to make the modification. So that region can complementary bind with the DNA, and it can create the double stranded break, and this double stranded break, it is going to repair by the non-homologous end joining, So this is the two mechanism, the self hollow, non-homologous in the joining. In that mechanism, either some of the bases is going to remove or some of the bases is going to add. So in this mechanism, the gene disruption by the insertion or deletion, it can be possible. So in this way, we can do the gene knockout. And or if you want to create the mutation, it can be mutate. So it in this way the gene function is altered. And the second mechanism, the self hollow, is the homology directed repair. So if there is any mutation you want to make it correct, so you need the PCR product with the homologous region of the place where you want to make the correction. So this double stranded DNA product you can transform into the bacterial cell or cell, eukaryotic cell, where the cell have the CRISPR system and the guide RNA, and it can be recombined with the your target region and it can correct the gene mutation. So the correction is also possible. So they use for such type of correction in sickle cell anemia, muscular dystrophy, because thalassemia, because if the very single gene based mutation is called the disease. And with application, you will see you can clear the model for the disease, you can correct the defective gene, you can control the virulence gene, eradicate the virus, bacteria, fungus. So it has implemented a lot. The second way of using the CRISPR system is the dCas9. So this is the another version of the Cas9. So dCas9 means dead Cas9. So in the Cas9 protein, the 10 position and in the protein 10 position and the 840 position there is active region. So if you make this mutation in that region, the Cas9 become the dead or inactive. So but it retained the binding ability, but it lost the double stranded break ability. So in this way, you can design the guide RNA and the whole complex can bind with the target region. So in that target region, it can refresh the transcription initiation. So it can bind with the nearby of the region where the ribose RNA polymerase can bind. So it and you will see there is no transcription at all. So R it can bind the transcription elongation location. So RNA polymerase it is going to transcribe the DNA, but it can bind there the whole complex of the dCas9 and the guide RNA. So it will not allow the RNA polymerase can move. So in this way, it can stop the transcription elongation. Are some of the activator molecule like the BP16 in eukaryotic system, BP64, this is the protein, or omega, which is present in the E. coli. If you fuse this type of the protein with the dCas9 with the guide RNA in the presence, so it can activate the ribose RNA polymerase activity and it can make the boosting of the gene express transcription label and it can activate the transcription. If you want to see the imaging of the different chromosome where the gene is present, maybe 16 chromosome number, 5 chromosome number, so it can be used to fuse the green fluorescent protein gene with the dCas9. So in this way, it can bind there, and you can use the microscopy to find out the location of the gene on a particular chromosome. So these are the few application of the CRISPR interference we know. And the very short span of the time, the professor Carpentier and the Dorna received the Nobel Prize for the discovery and the development of the CRISPR-Cas technology. So based on the CRISPR interference technology, we designed the autonomous computing machine where we use the AND gate logic function and we go for the making 27 AND gate, NOT gate and the S gate, because in order to make this system, we need the different kinds of the logic gate. So based on that, we characterize the autonomous system, and here you can see we initially do the experiment where constitutive expression of the small guide RNA, and we made the another plasmid that express the GFP under the control of the T7 promoter, and the DCAS9 express under the consecutive weak constitutive promoter, because the DCAS9 is toxic to the cell, and if you express more, then a little burden is going and cell is not happy to grow. And here the minus and the plus sign, it indicates that if there is no guide RNA present, the fluorescent is high. If the guide RNA is present, the fluorescent is down, because it's used the CRISPR interference. So the GFP you cannot see very high. So all the plus sign indicate that we design the guide RNA that can replace the GFP 99%, and it's almost nothing. So with this way, we design 20 different type of the guide RNA, and we characterize to each other. And we check also the orthogonality of the different guide RNA. So none of the guide RNA is matching together, and we were able to do this experiment. Here we tested the circuits based on the, here you will see the orthogonality, where is point 0.1 is the maximum, and none of the guide RNA in our case was crosstalk, and any issues happen in this experiment. So all are the independent and orthogonal, because we wanted none of the guide RNA to show any similarities. Now we use the different Cas9, that is called the Cas13, because the Cas9 is only breaking the double stranded DNA, but the Cas13 have the ability, it can degrade the RNA and the DNA both. So if you want to use as a diagnostic purpose for the Cas, then we use the Cas13. And you know, the if you have the RNA molecule, then we need to do for the RT-RPA. RPA means recombinase polymerase amplification. So this is the another type of the method, uh, apart from the polymerase chain reaction PCR method. So it can be done within a 10 minute, and you will find the intensity and concentration what you are doing in 2 hour or 3 hour in the PCR machine. So it is very fast polymerase, and it, you can see the amplification is very fast. If you have the DNA molecule for the detection purpose, you can use the recombinase polymerase amplification to enrich the DNA number, DNA concentration, and once you have enough DNA, either from the RNA source or from the DNA sources, we need to do the T7 transcription to make more mRNA molecule, and we have the our guide RNA, we have the Cas13, we have the fluorescent molecule. So all we need to put together, and we do the cleavage reporter assays. It can be monitored through the fluorescent microscopy, or it can be use the nano gold based material to see on the lateral flow. So we are working in this area to develop the diagnostic kit based on the CRISPR Cas13 system. So here we amplify the Cas13 gene, and we clone here the Cas13 in the PET28 vector, and we transform into the E. coli, and we have the clone for the Cas13 in the PET vector. Then we express the Cas13, and we induce with the IPTG molecule, and we found that Cas13 is expressed well. Because of the little toxicity of the Cas13, it is not expressed as what the other protein expressed, but we found the reasonable concentration of the Cas13 in our induced cell, and we purify the Cas13, and now we are making the using the nickel anti-achromatography column, and now we are building the lateral flow for the detection of the Klebsiella pneumoniae pathogen. using the Cas13 system. Then third work we are working on the metabolic engineering of the E. coli, and you know that all the cells we consider as the cell factory, because if you consider like the glucose metabolism, there is the glycolysis and the Krebs cycle, and in the Krebs cycle there is the all the organic acid is available there: citric acid, malic acid, fumaric acid, oxaloacidic. So all this acid we consider as the organic acid for the industrial application. Once the pathway is going to extend, there is the amino acid. So this amino acid we can consider again the industrial product. So you see the cell we consider as the cell factory. Whatever input you are going to give, the cell is going to process and give the output. And we face the number of the challenges in the metabolic engineering. First is the product toxicity. So the normal cell does not have enough tolerance power, and we need the very powerful strain, powerful cell that can tolerate once the production is high. So the wild type of the E. coli does not have enough tolerance to produce very high amount of the chemicals for the industrial application. In order to enhance the tolerance power, there are the number of the techniques has been used. One is the global transcription factor, or engineering of the export import pump. So once the molecule produce, it can pull out. So if the molecule is not inside, so it is not going to harm, it is not going to cause any toxicity effect. The third one, we can use the adaptive laboratory condition, where you can put the stress to the cell and it can produce the high amount of the your desired molecule. So it's like the gymnastic practice. So you need to do 2 3 months and you will see the high level of the tolerance of that organism. So here we use the evolutionary engineering, that is called the adaptive laboratory evolution, and we use the lambda system to inactivate the mute S gene. Mute S is the very important gene for the making any mismatch repair. So if you inactivate the mute S, then the repair mechanism is gone. So the genome is getting the mutation, and in this mutation, the beneficial mutation can give the tolerance to the cell. And we did subculturing of the cell, and for the 3 month, and we identify the number of the mutant it can give the higher tolerance. So it's you can select the organism which one is the fast growing. So in the industries of the especially the fermentation, there are the challenging parameters: temperature, salt, pH, and the solvent. So we chose four different parameters, which is very important in the biorefineries, and we were able to generate the mutants that can tolerance very high temperature. Here we saw the temperature is 46 degree, and our strain grow very nicely on the 46 degree. And even if you produce the acid, we need the strain that can tolerate the acid. So in E. coli, normally 6 to 7.2 is pH is okay for the cell, but we reduce the temperature and we identify the number of strain it can grow very nicely on the pH 4.5, pH 5, pH 5.5. And similarly, we use the butanol and the salt to create the mutant with the adaptive laboratory evolution to give the tolerance, and it can produce the high amount of the. And we extended the metabolic pathways for the lycopene. As you know, the lycopene is the important pigment for the antioxidant, and we constructed a vector that can be have the three missing gene which is not normally present in the E. coli. So we extended the lycopene pathways in the E. coli. That is the CRTEBI. So the three genes we construct under the control of the P-thiacy, which is the strong promoter, and we transform into the E. coli, and the E. coli we expect it can produce the lycopene, but it was not happen. And that time we use the multiplex automated genome engineering technique. So this is the technique where it's involve the DNA replication, and in the DNA replication, as we know, there is the leading strain and the lagging strain. And the lagging strain, it's giving the small segment that is called the Okazaki fragment. So we, any of the region, if you want to modify it, so we design the single oligonucleotide, and with the homologous region of where we want to make the changes, and we change the ribosome binding site of the DXX and the IPI, which is the rate limiting gene in the lycopene production pathways. And after the randomizing and the changing the ribosome binding site of this rate limiting gene, we will be able to produce the different concentration of the lycopene in the E. coli. So we successfully completed this work, and we found that our strain was able to produce the good amount and the different amount of the lycopene on the cell. And now we are working also on the biolysine, which is the color pigment. As you know that there are the number of the synthetic dyes is coming from the petrochemical, for the food, for the cosmetics, textile, and the pharmaceutical, for the printing. But the because of the depletion of the petrochemical and the toxicity and the non-renewable issues, so the global food color market is now 1.79 billion dollar, and the number of the synthetic dye are banned. So the global need for the natural and the safe, and now it has the 600 million dollar, and it's increasing every year. And now we need the natural pigment, which is cleaner, safer, and eco-friendly. So the organism, microorganism that can produce the biolysine is the Chromobacterium biolecium. So it has been used as an antimicrobial, antioxidant, anti-tumor. It can use for the food coloring, cosmetics, and the textile. But this natural organism has low productivity of the biolysine, and if you want to make any product for the industrial application, at least you need the 10 gram per liter for the industrial application. And this organism have also the coming with the class 2 pathogen, so there is the risk problem and the biosafety problem, and it's sometimes difficult to scale up for the industrial application. So we use the metabolic engineering along with the synthetic biology to take the biosynthetic pathways, which is present bacterium, and we want now to make these pathways into the E. coli. So all this violet color bio A, this is the 5 gene based cluster which we have already taken from the chromobacterium. We already amplify, so it's like the 8.5 kb of the gene, and now we are building the plasmid, and we want to extend the tryptophan native pathways which is present in the E. coli into the E. coli bacteria to produce the biolysine. So we are working on this area, and now we are constructing the biolysine whole cluster which have 8.5 kb, and once it is done, then we will be able to produce the biolysine in the E. coli. And we have already used successfully the process for the biolysine production using the very cheap carbon sources, and we also developed the method for the better extraction of the biolysine in the wild type of organism, and we have already filed the
கால்
patent for that
பண்ணியிருந்தீங்களே.
one. So apart from my scientific work, I am voluntarily working as a editorial board member
மிஸ்டர்,
of the associate editor of the
சரி ஓகே,
Microbial
அவங்க
Cell
முடிக்க போறாரு
Factory,
அவரு.
which is one of the very good journal in our area.
கவர்
frontier
பண்ணல நான் பாயிண்ட் நோட் பண்ணி
energy
வச்சிருக்கேனே.
review, frontier bioengineering and the biotechnology, frontier
சரி, ஓகே
microbiology,
சார்.
synthetic biology, which is published from the Oxford Group, and the scientific report. So this is the volunteer
அவர் முடிச்சுட்டு நான்
activity. I am
பேசிட்டேன்.
working for the growth of the science and technology in this area, and we
அவர் பேசி
learn a lot. So
ஆ
my interest is
ஆ
also because sometimes we are reading the book, but we are not getting the information what we
ஆ
are learning.
அவர்
So until now, we
ஆ
have published around 25 books on the different topics
ம்
more related with the synthetic biology, metabolic engineering, genome editing,
ஓகே
bioinformatics, metabolic pathways,
ஓகே
and
ஓகே ஓகே
macropeutics. So these are the my interest is coming in
ம்
this area to
ஓகே
help the
ஓகே
student researcher and the new one to accelerate and more better understanding how we can improve at our own level. So I acknowledge my student who really working hard to achieve the laboratory goals, and they are working. So too they receive the ICMR SRF fellowship because we have published a lot of paper, and that's why they receive the ICMR SRF. And so now we have laboratory. We are looking one more PhD student. So maybe 1 2 month I have already advertised the position. So we hope we can get the good candidate for our lab to accelerate. And I have the several international collaboration. Dr. Vinod Kumar from the Cranfield University. So he in UK. So he is helping for the downstream processing of our product, and Alfonso Jaramillo for the synthetic biology experiment. He is a professor in Warwick University, UK. And Professor Antonio Chagonas, he is also helping me for the phase therapy experiment. She is expert in the phase, and she is in Warwick University, UK. And for the biomedical and the COVID related work, we work together with Dr. Jin Deng Toi Chu. He is the director of the Biomedical Center in the Vietnam National